Okay. Yep. Yep. Okay. So to conclude our very engaging research session for this afternoon, we're going to hear from Dr. Vic Khurana, who will be discussing the future directions of MSA Coalition funded research. Dr. Vic Khurana is Chief of the Division of Movement Disorders at Brigham and Women's Hospital and Harvard Medical School. He sees MSA patients at his Parkinson, Ataxia, and MSA clinic at Brigham and Women's. And we're very pleased to have his expertise as a scientist and a clinician on our MSA Coalition Board of Directors, where he also acts as our scientific liaison to the Scientific Advisory Board. Please welcome Dr. Vic Khurana.
Thanks so much, Pam. Can you hear me okay through this? Okay, great. So it's just terrific to be here. This is the third time I've spoken at this amazing meeting. First time with a Mickey Mouse band on, but amazing venue, amazing talks. It's a real privilege to be here. So I think you've heard this morning, this afternoon with Professor Wenning's talk, that this is a time for optimism in MSA. It is unbelievable how many connections are being made to us in academia from industry partners who are interested in getting drugs or different biological therapeutics into patients for this disease. It's unprecedented, and it should give us a lot of hope. But there is a lot to be done, and the MSA Coalition is proud of the work that's already been done, but aware that there is a lot that is going to need to be done to tackle this difficult and rare disease. So last night at the beautiful candle lighting ceremony, I lit a candle too. I don't have a family member who's had MSA, so I don't really know, even though I'm a physician who sees patients with this disease, I don't really know what any of you are really going through. However, I do remember very fondly these patients that I see for many years, many of us clinicians do, and I lit a candle last night for some of them, and in particular, I was thinking of a patient, Miss S, who's from the New England area. Now, a few weeks ago, she ended her fight with MSA, but thanks to the incredible generosity of her and her family, our association with her is now not just through memories, but through actively studying her in our laboratory. And I'm going to end this talk by coming back to her and saying that it's through the generous donation of her biospecimens and eventually her brain that we believe that we are on the cusp of making important discoveries about this disease. So this is a story that is familiar to you. She first started reporting imbalance in 2009. I saw her in 2013 at the Mass General Hospital, where at that point she had no diagnosis. And I know that resonates with many of you, that for years you can go without a diagnosis. She had been told that she had Lyme disease, and she was being placed on antibiotics every day, intravenous antibiotics, and she did not. She had a different disease. So over the course of the next 5 years, she progressively deteriorated her balance and speech. She had autonomic changes that you've heard so much about and that you've experienced yourselves. She had urgency, and she acted out her dreams. In 2019, she transitioned to hospice care, and a few weeks ago, she passed away. But I have to tell you that until the very end, she had tremendous spirit, and she was literally cracking jokes with me in early August. An amazing person, and it is because of patients like this that we stay in the game of doing research and of continuing to come to clinic. But we need to do better, of course. We need to do better for patients like this and like you. And the way we do better is we engage in research and we collaborate. We collaborate with our patients collaborate with their caregivers, we collaborate with scientists, and we all work together as a team. Nothing will happen without this collective effort for any disease, but especially for a rare disease like MSA. So this is a great time not just for MSA Research, but also for the MSA Coalition. Thanks to tremendous support, and of course it's well-deserved. I'm officially part of the MSA Coalition, but in the 30-year history, I've just joined at the very tail end, and I can tell you this is the most inspiring group of people I've ever worked with. And it's because of their hard work that they have engendered so much support from the community, and through larger donations, they are able to now be more ambitious with what they support. So today, in the next few minutes, I'm going to tell you a little bit about these research directions. Okay, so where do we go to figure out what our directions should be? A few years ago in Las Vegas, of all places, on Halloween of all times, a group of researchers from all around the world met together and decided on some of the biggest priorities facing the field that are barriers to us treating patients more effectively. And so it is through working with those colleagues, through countless conversations over the last few years, that we've started to develop ideas about a coherent collaborative strategy that may work moving forward. So the first issue is what causes MSA. You've heard it this afternoon. Short of bad luck, we don't know why this happens to patients. We don't understand why it is that genetic studies have not really been fruitful. We don't fully understand the environmental factors. It's critically important for us to answer this question. The second is, where does this pathology start? You've heard about different cells in the brain, neurons, oligodendrocytes, astrocytes, microglia. These are different cells in the brain. But where does this disease start? We still don't know. And if you want to study something in the lab, you need what's called a model. Now, none of the therapies coming at us right now would have been possible without the extraordinary models, such as in mice, that have been made by labs like the Stefanova lab and the Venning lab. But we do need additional models, and we may be able to create human models, for example, from human stem cells now, that could be very, very valuable in understanding this disease. What drives progression and how do we measure it? You heard this in the talk before on the global MSA registry, that it's so important for us to track patients, to know who they are, to know what they go through, but to also have biospecimens so that we can diagnose this disease early, and if we engage in a clinical trial, we have something to follow. Otherwise, how would we know that the drug or the intervention is beneficial? And finally, of course, we need to know how to treat it. Not just therapies that prevent the disease and slow it down, but therapies that improve quality of life and improve the symptoms of our patients. So these are all critical questions that need to be answered, and they need to be answered in a systematic way. Now, what you've heard is that in previous years, the Coalition has supported many groups to do wonderful work in their seed grant program. And that means that the Coalition has been able to understand that there are the global landscape of who is doing what in this field, and how can people come together in groups to now engage in larger projects. And so at this point, the Coalition is thinking of not saying no to those types of individual seed grants, but to also start to support collaborations between different groups so that they can work together, because for a rare disease, this collaboration is essential. And so we're thrilled to be able to present to you ideas about creating collaborative international cores that are centered around some of these key questions. For example, Core G, looking at genetics. Dr. Dixon asked us the question, why is it that these genetic studies have failed? Could it have been that they were not powered enough with the numbers of patients? Could it be that there are private mutations in New England or in Arkansas or in Japan that cause this disease in small groups that you would never figure out in a population-based genetic analysis? These are all questions we need to answer. Environmental effects, we need to understand these more systematically. Can we create better models? That will be the job of the Core M, to create better ways in the laboratory to study this disease. A Core B, this initiative being driven by Dr. Claassen, can we do better at more systematically across different centres generating biomarkers and biospecimens? How do we leverage the amazing work that was done with the global MSA registry to incorporate biological tissue and specimens into that database? And of course, we would not be closing the door to any unique projects that come through the door that seek to improve the patient's quality of life or to create new ideas for therapeutics. So the idea basically is that number of sites individually have been supported, but these sites should now start to work together in collaboration. And so while previously you can think of seed grants as seeding one group, these are seed grants that would seed multiple groups. These obviously need resources, and so these resources are now available to begin this sort of program. Now, what are the underlying tenets, what are the rules of this game? The first is that we need to use existing data. The MSA Coalition is still a small organization, and it's not going to tackle this disease alone. It also means that we should use what's already out there and more efficiently use data that's already been generated in different groups. So in other words, don't reinvent the wheel. We need to share data. So we cannot exclude any groups. If anyone, whether that's in academia or industry, has a good idea, they should be welcome to the table. That's a critical aspect of this. And we have to build sustainable funding. The coalition can start a process, it can catalyze a process, but it is going to need support from the community, from industry, from government in order to create a sustainable framework. So what are the funding mechanisms here? Well, there are three basic mechanisms to fund this. There are coalition-initiated cores, and so initially these ideas come from the board of directors, but it's critical that an external group vets these ideas, and we have a very impressive board, a scientific advisory board, that objectively looks at all of this and decides whether or not the proposal makes sense. But then you need this to be inclusive, and so the second part of this is absolutely critical, and that is a core expansion grant. If someone around the world says, well, that sounds interesting, you're doing a genetics project, I'm doing a small genetics project, can I join? The answer is absolutely yes. So that is a critical part of expanding the cause. And the third, as I've mentioned before, is to continue the very successful seed project grant mechanism, because as Professor Wenning has showed this morning, it's been so successful so far that we would not want to stop that kind of progress and be too determined about what's going to work or not. We need original, bold, new ideas that we haven't thought of. So where are we at with these cores? So Core G, the Genomic and Computational Analysis Core, this has been coalition initiated, and it is aiming to begin in 2019. I'm delighted to say that this week the Scientific Advisory Board vetted this project thoroughly. We offered a detailed proposal and agreed to fund this core. And so we're hoping that in 2019 this begins. You've heard a number of times this morning about Dr. Sonia Schultz's great work at the NIH supporting 1,000 genomes, the sequencing of 1,000 MSA patient genomes, which importantly is in addition to many thousands of genomes for Parkinson's disease and dementia with Lewy bodies, and this is going to be a tremendous, potentially game-changing resource for the field. The coalition wants to support that initiative. Second, we want to understand environmental exposures, but this is just the beginning for MSA. If Parkinson's disease is anything to go by, it's going to take us a long time before we understand and can categorize environmental exposures in a way that could be useful for laboratory research. But what the coalition wants to do is engage with some of the world authorities on Parkinson's disease epidemiology and exposure, and have them help us develop a systematic questionnaire that should go to any center that's interested in MSA research, so that we can systematically gather across all of what we fund and beyond this kind of information from our patients. The MCOR, which is to advance human, cellular, and animal models. We exist in a time where we can grow even little brains in the dish from our patients. It's an incredible time for stem cell research. Can we leverage this technology? This core is hoping to get off the ground in 2020. Core B, biospecimens and biomarkers. Potentially the most important of all of these, actively being planned right now. Again, hope for a kickoff in 2020. So I'm going to return back to our individual site at Harvard Medical School, at Brigham and Women's Hospital, and I'm going to close this story by just giving you a little example of how it is that groups by themselves cannot do this, and they need to work together. So at our individual site, we have a building here, the Building for Transformative Medicine, where we see patients and do imaging in the ground floors. We have MRI and nuclear medicine capabilities. And above that, we then have a tower of labs where research gets done. This is a great environment to bring researchers together with clinicians and with patients. When patients enter our clinic, just as you've heard with many of the clinics here at University of Florida, Vanderbilt, other places, there are systematic programs to analyze patients. So, for example, our patients undergo brain imaging, they give biospecimens, they donate brains. But we also take skin biopsies from our patients or from their blood. We can generate stem cells, we can create brain cells from our patients, and even these mini brains. It all starts with the work that we do in our clinic, and so these amazing individuals are some of the group that I work with every day to evaluate our patients with MSA. And it was exactly in this clinical setup that Miss S, who I introduced at the beginning, was seen. Now, she was one of our cohort, and we were collecting her samples and building up this information bank. And it would have potentially stayed like that if I hadn't been contacted by a human geneticist. And this highlights the power of collaboration. So this geneticist, Matt Huntleman, contacted me because he felt that looking for common changes in genes may not be the way to look at MSA genetics. We should study MSA in families. So that's called a trio study, where you can sequence parents and you can sequence a patient with MSA and sometimes their siblings as well. So he sequenced about 20 different families where we could get this information. So a relatively small study. But he got back to me and he said that the patient that I was following, Miss S, was rather remarkable. She had parents who were nearly 100 years old at the time, where we could get sequencing from them, and she also had 6 brothers and sisters, so a very large family with 2 parents who were alive. And what he pointed out to me was that she was the only one of her 7 siblings that carried 2 gene mutations that she had inherited, one from her mother and one from her father. And he told me what this gene was, and he wondered if I knew anything about it. And it turns out that the MSA Coalition, as you heard before, had supported me to study how it is that alpha-synuclein systematically interacts with genes in cells. And it turns out that the gene he told me about in one of my patients was a gene that I had actually previously identified and published on in a study that had been supported by the MSA Coalition. Moreover, this was a rare gene, something that was not studied by many people. We found a lab nearby at Harvard Medical School that was actually studying this gene. It's involved in metabolism. And so suddenly, a finding that could have just laid dormant in my lab was activated through a very rich collaboration with a genetics group and also with a nearby lab at our medical school. But of course, could this be only a change relevant to my patient? How relevant could this be to other patients with MSA? So one of the things we did was we reached out to Professor Shoji Tsuji at the University of Tokyo, who follows one of the largest cohorts of MSA anywhere in the world. As you heard from Dennis Dixon, they often have a cerebellar type MSA, which is exactly what my patient had. So we asked Professor Tsuji if he could interrogate the genetics that he has done with the 400 genes that we know interact with alpha-synuclein, the protein that you've all heard about. And it turns out the number one hit on the list of his 600 patients was the same gene. So of the 400 genes that we studied that were picked up independently by Dr. Huntelman, he picked this out as the number one gene in his cohort as well. And I don't think this happens by chance. So we think this is a very, very interesting finding that's worth pursuing. It's early days, but we think it's very exciting. This would not have happened without collaboration. It's exactly the kind of collaboration I think we're trying to support. So now this research goes straight back into my laboratory, which is exactly located in this corner here, and I work with a team of very talented students and postdoctoral fellows who are pictured here, and thanks to incredibly generous donation that made the Barbara Bloom Ranson Fund. Barbara Bloom Ranson was a patient with MSA. We were able to start to dig deeper into what this gene may be doing. And we began a program that's called the Match Brain to Stem Cell Program, in which patients who have either donated their spinal fluid or their brain, we make culture brain cells from them in the laboratory so we can create new types of stem cell models for this disease. So in this summary diagram here, you can see that we can take a small skin biopsy from patients, we can turn them into stem cells, and we can generate brain cells and mini brain organoids from these patients. If a patient donates their spinal fluid or donates their brain if they've passed away, we can capture the toxic alpha-synuclein protein from that, and we can introduce them into brain cells that are engineered to show us the aggregation and accumulation of that protein. And so here you can see the process here, the skin biopsy or spinal fluid. This is an example of the brain pathology that we see that you saw from Dr. Dixon's work. This is showing the glial inclusions of alpha-synuclein. Here are skin cells that are turning slowly into embryonic stem cells, rather remarkable technology, and we can turn them into stem cell colonies, and from those we can generate brain cells from our patients. So you can see these are brain cells that we're growing from a patient. You can see these green dots here. We've engineered the brain cells so that they can show us the aggregation of the alpha-synuclein protein. In collaboration with Nikon in Japan, we are developing techniques so that we can track each of these individual brain cells in the dish, and we can watch the formation and dissolution of these aggregates and learn a lot more about the biology of the different forms of alpha-synuclein that cause this disease. And we've engineered these so that we can fairly quickly turn these flat neurons into three-dimensional brain organoids. And so you can see here, this is a three-dimensional structure that we've made from a patient. It looks very much like a mini brain, and we can inject alpha-synuclein into it, and we can watch the spread of alpha-synuclein in three-dimensional space. So this is a remarkable time where we can study someone's genes in their own cells even when they're living, because we can make brain cells and these mini brain organoids from our patients. And so I really hope that these kinds of technologies bring you hope. They bring me hope as a physician scientist, and I hope together we can do something. This story obviously has to be continued. We need a lot more to get to the bottom of this, and we're hoping that with your support we can do so. So just finally, I want to thank the incredible people that are colleagues with me on the Coalition Research Committee. A special shout out to Pam Bauer, who is just a unique individual without whose tireless efforts none of this research would be possible. So thank you, Pam. And none of this is also possible without having a stellar scientific advisory board who you can see here. And of course, last but first and foremost, I want to thank you, the patients and caregivers, because without your help, none of this would happen. So thank you very much.
Thank you so much, Vic, and I'm so proud of this program, and I'm really looking forward to the next few years. So now we'll move to the questions and answers session. I believe a lot of questions have already been sent to the front. So who wants to take the first question?
Just start with the question?
Yeah.
So we received a number of questions here. One is, what are the most promising biomarkers currently for diagnosis and disease progression? That's a difficult one. I could maybe direct that to Daniel. You want to answer this one?
So the biomarkers right now that are potentially being used for MSA, one is brain imaging. So if you look at MRIs of brains and certain patterns of the way the brains change and certain collections of things like iron or changes in white matter integrity, where that's located, that has potential to be a good biomarker, not only for is it MSA, but also how is the disease progressing and how severe is the disease. Another one that's being used is different biofluid markers, and these have been really tried in other neurodegenerative diseases, but things like there's these things called neurofilament light chain, NFL, is one potential biomarker of disease. And then there are other ways to potentially look at alpha-synuclein, which have been used in Parkinson's disease recently to diagnose and potentially follow the severity of disease. And I think the third one that I would say hasn't come to fruition, but really I think needs to be explored is the clinical progression. How do we quantify how much disease a person has and how it's progressing? And I think that's really one of the goals that the biomarker and clinical follow-up of patients program has, is can we actually figure out how to determine how a patient is feeling, how severe they are, and how those feelings and symptoms change over time. Because if you had a therapy that could show was working, you really want to show that it affects patient lives rather than just, you know, a clinician's exam. And I think that's really the hope, that in terms of clinical biomarkers, we can get a lot of momentum, especially with these kind of collaborative clinical assessments that were referred to. Is that enough?
I think there are very many questions. I'm not sure whether we can get through all of them. There is a question on the MSA mouse models that we have been using to screen new drugs that might help in MSA. And the question is how that really works in a mouse model. So just to explain how we do that in a nutshell, we are basically overexpressing the human synuclein in the glial cells in these mice. So basically, when we look at these mouse brains, what we see is exactly the same types of synuclein inclusions in glia that we see in the human MSA brain. And so what we do then is that we just follow up these mice that have these synuclein buildups in the brain. And interestingly, as these mice get older, they develop spontaneously, if you like, they develop the Parkinson phenotype of MSA. They don't develop the cerebellar phenotype, they develop the Parkinson phenotype. So to us, this indicates that the synuclein inclusions in MSA, if they are really the primary event that happens in these brains, they are able to trigger the Parkinson-like pathology. And we also have evidence that they attack the autonomic nerves in these MSA mouse brains. So I think that's why these models have been very valuable to us. The second question is about dementia in MSA. I think I can probably reassure you by saying that dementia is exceedingly rare in MSA. Some patients might have mild problems with what we call frontal lobe function, which means that you sort of shift from one exercise to the other, from one task to the other. There may be some delays with that, but this is really nowhere near the type of dementia that we see in Parkinson's dementia or dementia with Lewy bodies. So why dementia is so uncommon in MSA is unclear. But certainly this is not a kind of major issue, I would say. Then there's a question whether it's possible at some stage to reverse all this toxic synuclein buildup in nerve cells in MSA patients. Obviously, this has always been touched upon in the presentations today. It's very important to identify MSA as early as possible, and there's evidence that you can even diagnose MSA in the pre-motor phase by autonomic issues or REM sleep behavior disorder. And interestingly, MSA patients in these very, very early stages, they have a preserved sense of smell. Olfaction, which is destroyed in Parkinson's disease, is preserved in MSA. So by kind of this pattern recognition, you may be able to diagnose MSA very, very early on. And this is part of also of the ongoing natural history study that Dr. Kaufmann presented. The last one, before I hand over to the colleagues, is about supplements and vitamins and herbs as part of the treatment. There's really no evidence to support that, but in my clinical practice, what I recommend to patients with MSA, particularly when they are kind of early in the disease, I recommend complex vitamin B types of supplements. There's one that we use in Austria mainly, and I recommend a kind of pulse of that, like for 2 months and then stop for 2 months, start again for 2 months. And I also recommend to combine that with coenzyme Q10 supplements because there's this evidence from the Japanese literature that this might work in the cerebellar presentations of MSA. So with that, I hand over to the others.
So the first question I have directed for me is what criteria are required to be the high-risk subset in the natural history study? So if you recall, we are following a number of patients that we believe are high risk of developing MSA in the future. We are including patients that have REM sleep behavior disorder, which is dream reenactment, acting out dreams. So many times these patients may shout, scream, get out of bed, or react to vivid dreams when they should be still. And we know that of that, and some patients will develop MSA, and that may be the first sign that they have. We are also following patients that have neurogenic orthostatic hypotension, which is a fall in blood pressure on standing and low blood pressure in the upright position that's caused by a problem with the nervous system. And what we see in our cohort is that if we take those patients, around 8% will develop multiple system atrophy within 2 to 3 years. And then if you take what we were able to do, which goes back to finding MSA sooner, is see, like Gregor Wenning said, is that preserved sense of smell, a high incidence of bladder abnormalities or a need for treatment of bladder and GI abnormalities, and as well as some autonomic biomarkers like a slightly faster heart rate or signs that the sympathetic nerves going to the blood vessels are intact may also be pointers that this might be early MSA, along with the patients having preserved sense of smell and no real cognitive abnormalities in the early stage. So that group of cohort of patients patients we are closely following to look at their trajectory to see how they evolve. The other question we have is, what's the role of head injuries in multiple system atrophy patients and in research, and are you finding a link? We've spoken a lot about what could be the potential factors that predispose multiple system atrophy. We've heard about genetics, and so far we have genetic studies which may be underpowered, but since we don't see any direct inherited traits, we are looking also in other factors with the possibility that genetics, our answer may lie in other areas. So in terms of head injuries, when you come into the natural history study with MSA, we will ask the clinician to collect a strong history from you, looking at family incidence of dementia, things like head injuries, smoking, all the types of history that you would normally get, to see within the cohort if we can pull out some of the environmental factors that might be contributing. So I'm going to pass the microphone now to Dr. Clausen.
I feel like I've talked too much already. Okay, we'll go through quick ones. Do clinical trials have fees? No. If someone charges you to be in a clinical trial, call the police. Are clinical trials vetted on clinicaltrials.gov? I think they're usually, it's kind of there's no one that tells you whether it's a good trial to be in or a bad trial to be in. You can sometimes look at the funding sources to get a sense of, who's advocating for it. For instance, if it's funded by the National Institute of Health, it means that it's been peer-reviewed and that it's gone through quite a number of different levels of review. So they're not exactly, vetted for how good or bad they are, but they're just there. And you can also look at some of the outcome measures to kind of get a sense for how things are.
Daniel, can I just jump in there?
Yeah.
Someone had mentioned, Gregor had mentioned stem cell trials before, and at least until recently, even some of these privately kind of more dubious, doubtful stem cell trials were actually listed on clinicaltrials.gov. So be careful; it's not so well curated.
I'm kind of more interested to hear what Dennis has to say, but I'll do a couple more. Can you combine being in an MSA symptom reduction trial and a disease-modifying trial at the same time? Great question. When I talked about inclusion and exclusion criteria, oftentimes exclusion criteria include current enrollment in a clinical trial. And so I think most of the time, like I would say 99% of the time, you can't be in two trials at once, with the exclusion being if you're in an observational study. Oftentimes you can be in a trial and you can still stay in the observational study. But the protocol should have very clear guidelines as to whether or not you can be in another trial. Generally speaking, if you're getting a therapeutic intervention, you wouldn't want to be in another one. And you want to do one more? All right. I know you want to say stuff. All right, you're going to have the answer for us here. Do stage 3 trials benefit patients, or are they more for the benefit of science? Absolutely, they benefit patients. This is the question for stage 3. If I see you in my clinic and I diagnose you with MSA and I give you this therapy, does it make a difference? That's what stage 3 trials do. They replicate real-world enrollment and therapeutic trials to see if it works or not. And I think we generally guide our practice based on the trials in terms of how they're run, who's in them, and the outcome measures. So I think they're very important for patients. Last one. MSA diagnosis is a risk for clinical failure. Definitely. I assume that's for clinical trial failure, especially if drug target is specific for MSA. Any ideas to reduce this risk? I think one of the goals of like natural history study and in particular is to try and understand what are the clinical predictors of MSA. There are certain diseases that we're quite good at diagnosing based on certain clinical phenomena. I think with MSA, sometimes we have a little bit harder time, but things like imaging markers, things like clinical phenomena like, say, sense of smell or orthostatic hypotension, how much a person has, they're generally useful in terms of helping us to kind of more specifically diagnose MSA. So I think, yes, that's true, but I think we're doing much better at knowing which clinical features help us really narrow down on MSA versus something else.
Okay, so there's a question about MSAC and MSAP. Can a single individual have both diagnoses? And I guess I want to just frame this question in the terms of what's a clinical diagnosis and what's a pathologic diagnosis. So the clinical diagnosis is MSA-C. You have a clinical syndrome with cerebellar predominant clinical features. MSA-P is MSA with predominant Parkinsonian, atypical Parkinsonian. When we look at it at pathology, we talk about olivopontocerebellar atrophy or striatonigral degeneration. And they correlate pretty well. So striatonigral is usually an MSA-P clinical presentation, and olivopontocerebellar atrophy is usually an MSA-C clinical presentation. But in fact, the pathology in an MSA-C patient, there's pathology in the striatonigral system. In an MSA-P system, they always have pathology in the cerebellum, the pons, and the olive. It's the thing that determines which clinical presentation the patient has is where the neuronal damage is the worst. So MSA patients have pathology in both systems. Everybody has in both. They have olivopontocerebellar and striatonigral. But the degree of neuronal loss may be greater in one or the other. And the neuronal loss determines the clinical syndrome. So it's probably still not clear. But it's basically, think about the pathology being everywhere in terms of, especially the glial pathology, but where the neuronal loss varies. And I'll have to say, in some patients, especially at the end stage of the disease, it's not always easy to make a distinction in terms of OPCA and striatonigral degeneration because the patient has severe pathology in all systems. And we think of that as it's not a very good term, but it's sometimes referred to as end-stage disease, when basically all the systems are affected. And then there was a question about can a person do a brain donation if they have another clinical syndrome or pathologic clinical syndrome, I guess, because they don't have pathology. So, for example, if you had MSA, but you also had clinical features of, say, ALS. And absolutely, those are the patients that can be very informative if, in fact, they have two separate neurodegenerative diseases. And in MSA patients, for some reason, tend to be relatively pure in terms of Alzheimer's pathology. I showed in the table, I didn't emphasize it, but if you look at a Parkinson patient, a Lewy body dementia patient, they very frequently have comorbid Alzheimer's pathology. MSA patients almost never have comorbid Alzheimer's pathology. So if you, in fact, did have an MSA patient that had also Alzheimer's or ALS, that would be a very informative type of patient to study, because it might suggest some link between the different neurodegenerative disease processes.
Okay, so I have a question, and one question here is actually in two parts. So actually, Dr. Dixon may be a good person to weigh in on one of these. Someone has been interested in iron accumulation that was mentioned because a couple of the potential therapies are involved in reducing iron accumulation. And so the first part of this question is could you use phlebotomy, which is, just removing blood to treat these diseases? It's an interesting thought. It is used for certain diseases like hemochromatosis. It's been proposed for neurodegenerative diseases, but because of the compliance with phlebotomy, the need to take blood, chronically, people have generally preferred using drugs to reduce iron, so-called iron chelators, and the iron chelators that have been typically used for diseases like hemochromatosis do not cross the blood-brain barrier. So they wouldn't be effective for a disease like MSA, but that's why companies like Prana, for example, that Professor Venning mentioned, are designing brain-specific agents that can reduce iron. But the second part of this question, where it would be great to have Dr. Dixon's input also, is whether we should screen for hereditary hemochromatosis in patients with neurodegenerative diseases. It traditionally, I think, has been thought that hemochromatosis does not really cause cause, a degenerative brain disease. Although I believe some of that data is a little controversial, and there have been studies suggesting that there are changes, certainly at the cellular level in patients with hemochromatosis. But I don't know if you want to comment any more on that.
If the iron that accumulates in MSA doesn't seem to be a primary disease process, it seems to be late. And it could be as neurons die or the reactive changes in the support cells, the glial cells, that there's accumulation of iron. In terms of the hemochromatosis, there's almost no neurodegenerative pathology seen in hemochromatosis, and if there is iron deposition, it's an exaggeration of the iron that accumulates usually around blood vessels in certain parts of the brain that are different brain regions from those that are affected in MSA. So it's the basal ganglia, but it's in the globus pallidus, whereas in MSA, the pathology is in the putamen and the caudate. So it's anatomically, it's like real estate. These are different plots of land. So they're different disease processes. So screening for hemochromatosis would not be really very useful at all.
Thanks so much. And the last question was, can we manufacture dopamine-producing neurons from skin cells? Yes, absolutely. And so you can turn skin cells into these induced pluripotent stem cells, these embryonic stem cell-like cells that I mentioned, and there are good protocols to efficiently make midbrain type dopamine secreting neurons from these cells. And so there are a number of groups and companies that are testing this for Parkinson's disease. Now, Parkinson's disease may be a potential target for such a therapy because the loss of those cells is really profound, and it's a central part of the symptomatology of the disease, the loss of those neurons. Even in Parkinson's disease, though, many different systems of the brain and nervous system are affected. So whether just replacing one type of cell is a good therapy, there's pros and cons for thinking about that, and especially in a disease like multiple system atrophy, where, as you've heard today from the pathology, there are many different systems in the brain and outside the brain that are affected. The usefulness of just transplanting one type of cell back into the brain would be questionable. But the simple answer is that yes, you can make these cells, and yes, they could theoretically be used for transplantation, and these are being tested right now.
So I have a few more questions regarding therapy also. So somebody asks about high-intensity exercise for progression of MSA. I usually tell my patients it should not be high intensity. They should not get exhausted with these exercises. It should be like moderately, exercising moderately and feeling good with the exercise. So I mentioned to you the study that we've done with physiotherapy focused on gait disorder in MSA, where we saw like 20% improvements with the sensor-based technologies, these sensors hooked onto the shoe, and so we have some parameters there that tell us something is going on, and the patients felt well with that. So I would say no to high-intensity exercise, but moderate exercise would be good. And then another question, which is not so easy to address, which is the number one drug to change the disease course in MSA? I tried to highlight all the different kind of drugs in the pipeline right now. To me personally, I think there are 3 or 4 that maybe are particularly interesting. One is trying to influence neuroinflammation, and this is the ongoing MSTAR trial by Biohave, and that is interesting, an interesting concept. Then also interventions based on synuclein stopping to synuclein from aggregating. So I mentioned immunization, I mentioned synuclein antibodies, and there's also, which I didn't mention, there's also an activity now ongoing using antisense oligonucleotides trying to reduce the synuclein levels in MSA patients. So that's interesting. And maybe there is also the approach of small molecules, and I mentioned the ANLI 138B that's been so successful to reduce protein aggregation, including synuclein, in various models. There is one question about placebo effect in clinical trials. We all know from Parkinson's disease this can be substantial, up to 40% in some of the Parkinson's disease trials. There's nothing known about this in MSA. My guess is that in the absence of symptomatically effective interventions, maybe the placebo effect would be substantially lower. There is one question about MIBG scintigraphy in MSA. Is that useful? Just to explain to you, this is a diagnostic test that measures the integrity of sympathetic fibers in the heart muscle. And interestingly, in Parkinson's disease, there is a complete denervation of the heart muscle using this technique, MIBG scintigraphy. And in MSA, in most MSA patients, not all of them, but in most, there is a preserved cardiac innervation. So basically, it can provide useful information, but there are MSA patients who have pathology in the heart muscle as well. So sometimes it can be difficult. The last one is about, again, stem cells. We've talked about that already. I think right now I'm only aware of that one academic protocol from the Mayo Clinic where they have submitted that to NIH, where they would like to establish whether lumbar or whether mesenchymal stem cells delivered by lumbar puncture could modify the disease in MSA. So, but they haven't got approval for that. So that would be very interesting to follow.
So the question I have is about registries, and it's how is the NYU study you talked about different from CORDS? And CORD is the Coordination of Rare Diseases at Stanford. There are many different registries, because there are many different rare diseases, right? So there are multiple different rare diseases, which when you put all the rare diseases together, it affects a substantial number of people. Unlike interventional trials, is when you're trying something to see if it makes you better, you can participate in different registries in which you're just providing your information. So our specific registry for MSA is focused on MSA. So we're asking questions specific for MSA that may or may not apply for other rare diseases, but we are specifically looking at what may apply to MSA. So the short answer to this is yes, you can join other registries, but consider also giving your information to registries that are specific about your disease, because the other factor of that is that if you provide your information within a contact registry, if there is a treatment or if there is a local support group that is applicable to you, you can then get the information because it will come back to you. So there may be advantages of participating in a registry that's for all rare diseases, of course, to know the impact collectively of rare diseases, but there are also distinct advantages on participating in one that's specific for your disease.
So this question's from the internet. Basically, the question is discussing clinical team approach, and this individual is running into a challenge that the team approach is being restricted through Medicare rules on in-home care or outpatient. So I think for all of us that treat MSA, I think all of us enjoy the interdisciplinary team approach much better than just one person running the show, because everyone has different expertise. And Medicare shouldn't stop that team approach. sometimes it's true that rare diseases, the clinician, like the physical therapist or occupational therapist, may not know much about MSA, but part of what the team approach does is that everyone kind of educates each other. And so with our therapists, sometimes we do crash courses on MSA, and we'll have meetings with them and talk about kind of the issues we're struggling with, and we'll look at some of the literature that has been published on, you know, ways to tackle MSA therapeutically. So I wouldn't let Medicare hold you back from doing a team approach. Instead, I would look at it more as really encouraging the clinicians that you see to build those teams. When you demand that you want an interdisciplinary approach, people generally respond, and I think that's really one way to do it, is just kind of expect it. Do you have any questions? I think that's it. All right, well, thank you very much. We hit the witching hour.
Okay. Good job, everybody. Congratulations. So we have the option, if patients and caregivers would like to have another support session. We have facilitators willing to do that if you want to stay on a bit longer. But before we do go, I just want to acknowledge Cindy Romer for the great job she and the conference committee have done. And thank you very much for coming.
It's really good to see you, Dennis.
You're well.
Stand because
See if I saved his brain.